Okay, back here to continue with the urinary system. So we've now completed our knowledge on the digestive system with those 6 major roles. Now we progress to the urinary system, right? Urinary system is going to be involved in the water balance. It's also going to be involved in the buffering and removal of nitrogenous wastes, right? So we know that we're going to absorb nutrition from the digestive system and then defecate out what's non-absorbable, non-digestible. The urinary system is going to be involved in the filtration of blood, removal of water to regulate blood volume, and again, it has some other roles as well. So let's analyze here the urinary system. So the urinary system is pretty simple compared to some of the others, right? So the urinary system will be sort of beginning here at the kidneys, the paired ureters, the singular urinary bladder, and then the singular urethra. So again, what does it look like here? So we have our kidneys. So left and right kidneys is kind of like bean-shaped aspects here, left and right kidneys. We see this sort of yellowish tube. This is going to be the ureter. So the ureter leads down to the urinary bladder, ureter leading down to the urinary bladder. Urinary bladder then having that exit valve, which is then that exit tube, which is the urethra. So the kidney is actually the excretory organ, our body's primary excretory organ. Excretory as far as nitrogenous waste excretion, urine excretion, urine formation, urine excretion. Then the urine will be moved from the kidneys to the bladder. The bladder will store the urine until it's convenient to be urinated out, excreted out. And then the urethra will provide the passage for the actual urination process, or micturition as we're going to call it later. So what does a kidney do, right? Kidneys do a lot of things, but they're going to be involved in removal of toxins, removal of metabolic wastes, and excess ions from the blood. So because they do these 3 tasks, they're going to help to regulate blood volume. They're going to help to regulate chemical composition and have an impact also on blood pH. They also have an important endocrine role, right? So we talk about erythropoietin way back in the blood chapter. So the kidneys are the organs responsible for this release of the hormone EPO, erythropoietin, which will stimulate production of red blood cells. Renin is going to be another hormone that's sort of released by the kidneys to help the regulation of blood pressure and the kidney function itself. I use the term a little bit, peritoneal, retroperitoneal, when we talk about the colon. Well, we see that this is the peritoneal cavity. So the peritoneal cavity is where a lot of our sort of abdominal organs are held. The kidneys are sort of behind that. So they're behind, they're posterior to the peritoneal area, but they don't call it posterior peritoneal, they call it retroperitoneal. That's this location, right? The retroperitoneal aspect. Another idea of the internal anatomy of the kidneys, right? There's specific regions, and again, this is where I wish we had you in lab. I wish we could be actually looking at some stuff in lab, but we're going to have to make do with pictures here. So we have our renal cortex. Cortex is always like the peel, like the shell, right? The peel of the apple, the peel of the watermelon. The green part of the watermelon would be the cortex. The red part then being the actual medulla or medulla. So we're going to have the cortex, that surrounding part, the superficial region, the medulla, which is going to be sort of shaped in pyramids, sort of. You got to use your imagination a little bit, but pyramids, we'll see pictures. Lobes, papillas and a pelvis. And if you can imagine a pelvis, like where if you're going to do crunches or sit-ups, is that sort of flexion point, so that's the pelvis. We're going to have minor calyces and major calyces. So they kind of collect urine from minor calyces, which merge together to form the major calyces. And then finally the pelvis to the ureter. And again, I think this is going to help a lot more there, right? So here we have our renal cortex. So the cortex is all this superficial, thick, protective sort of area here. The renal cortex, like the watermelon peel, all around. And then we have, if you use your imagination a little bit, they call these renal pyramids. I've always thought they look more like seashells, but they don't call them renal seashells, they call them renal pyramids. But again, our renal pyramids made up of the medullary aspect here. So the cortex, the medulla. So there's our cortex and medulla. And then we get into this idea of the pelvis. So is that kind of part where there's kind of that little curve, that little sort of bend there. So the renal pelvis. What are the papilla? The papilla is just like the little tip, the little point of the pyramid. So again, some of the vocabulary that they're trying to explain here, it's just sometimes easier to see pictures and to see it all in word format there. But to kind of summarize what I'm going to get into next. So understand we're getting blood flow in, and blood flow will be sent in here to the sort of area to be filtered. We're going to filter, we're going to filter, we're going to filter, and then give blood back to the circulatory system. And all of the waste will then start to accumulate here in the minor calyx, eventually going to the major calyx, and then to the renal pelvis, and then out the ureter. So again, so we would form, we would filter the blood. The waste then would start to go to the minor calyx, aggregate into the major calyx, into the renal pelvis, and then out the ureter. So again, a little bit more detail here. You can see all the vascularization, all the blood vessels here. Right? These blood vessels are important because we're again bringing blood in to be filtered, and then we have to send out the filtered blood. And where does all the waste go? It doesn't go back into circulation. It goes into the minor calyx, minor calyx, minor calyx, minor calyx, minor calyx, merges into some of the major calyces. And then finally the renal pelvis into then the ureter. So it can be excreted out of the body to the bladder and then excreted out of the body. Okay, so the functional unit, the small little functional part of the urinary system. If you understand one nephron, if you understand how one of these works, just multiply it by a million, and you understand how the kidney works. A kidney is just one nephron magnified, multiplied by a million times, and that's what this thing is. It's just the same repetitive type of filtration device here, right? So the nephron, the structural and functional unit that forms the urine. So again, if we can understand one nephron, we understand how the kidney functions. There's going to be two parts of the nephron. One, the glomerulus, and two, the renal tubule. Renal tubule, also known as the Bowman's capsule. Right? When I learned it, I learned that the renal. I learned it as a Bowman's capsule, but it's the first part of that is part of the renal tubule here. So the glomerular capsule or the Bowman's capsule surrounds then that glomerulus. Okay, so if you can make sense of this, again, let's see. So this arterial blood supply. So we're coming in with arterial blood. And we're going to have this little cluster of arterioles. It's an arterially capillary, right? This is what we're going to call the glomerulus. And so we have all this mess of vessels, and then we have Bowman's capsule, or the glomerular capsule, that kind of wraps around it, sort of in all that sort of area. So from here, everything in yellow basically is part of the nephron, right? So we go through the process of going up and over. We're going to call this then the proximal convoluted tubule. Then the loop of Henle. And then up the distal convoluted tubule into the collecting ducts. Right? So all of that little picture that you see right there represents one nephron. And what is a kidney? It's just a million of these all working together simultaneously here. So we have then the idea of the nephron. We have, again, the glomerulus, the Bowman's capsule, glomerular capsule, proximal convoluted tubule, loop of Henle, distal convoluted tubule, and the collecting duct. So all of that being one nephron there. So the nephron is the glomerulus plus its associated glomerular capsule. So the glomerulus in red and its associated glomerular capsule. The glomerulus is kind of interesting, right? It's a neat anatomical design. So the glomerulus is the only aspect then that will it's an arterial circulation, and it's the only kind of capillary bed that's fed by an arterial and is drained by an arterial. Right? So again, that's the only part of our circulatory system that is going to be fed and drained by the arterial circulation here. So arterial blood in, glomerulus, arterial blood out. Right? So that's, again, unique there. The glomerulus is specialized for filtration. It's a relatively high pressure system, high for capillaries, right? So the nephron is a capillary bed, right? The nephron is, try to keep track of that, right? So the nephron is a capillary bed, and basically the nephron itself is going to be fed by arterial blood and drained by the arterial system as well. So specialized for filtration, assisted with high blood pressure. So again, if we can see, so afferent arriving, so afferent circulation coming in, it's red, there's no gas exchange here, and we leave via the red vessel here. So afferent arteriole, it's a capillary bed, and then we're draining with that efferent, exiting through the efferent arteriole. So all that is happening here. Inside, this would be glomerular capsule, Bowman's capsule, and that's then the glomerulus. Right? So glomerulus is a capillary bed, fed by arterial blood, drained by arterial blood. It's not drained by a venous circulation, is drained by arterial circulation. So we have all these fenestrated pores. So we have all these sort of network little filtration slits. So the whole point is we want to leak fluid out of the capillaries so it can be cleaned and sort of make it easy for things to leak out here, right? And then to be put in later as well. So the filtration membrane. So filtration membrane allows then fluid to leave the capillaries and go into the capsule, right? So here in yellow, we have two colors. So in pink is the blood vessel, the arterial fed glomerulus, and then in yellow is going to be part of the capsule itself. Right, so we're inside of the glomerular capsule. Now another term change. If I'm liquid in the blood vessel, I'm called plasma. If that liquid gets pushed out into the capsule, I am now termed as filtrate. So it's very similar to plasma, except it's not in the blood vessel. If you remember, if plasma gets pushed out into the extracellular space, we call it lymph. If liquid is, or I should say, if it's pushed out into extra space, it's extracellular fluid. Once extracellular fluid gets picked up by the lymphatic vessel, it's called lymph. If plasma is pushed out now into the glomerular capsule, we call it now filtrate, right? So it's basically the same kind of liquid, just in different parts of our anatomy. So I hope this is making sense, right? A lot of vocabulary here, but we're coming in with the afferent blood vessel, glomerular capsule, surrounding then the glomerulus, that capillary, and then that capillary leaves back into an arterial circulation, right? So here we have then the fluid, the liquid, the plasma being forced out into Bowman's capsule, into the glomerular capsule. And that liquid is going to travel down the proximal convoluted tubules, loop of Henle, distal convoluted tubules, and finally the collecting duct. Now, this is what we call a cortical nephron. Right? Cortical because it starts in the cortex and it dips down into the medulla. So this would actually dip down, the loop of Henle would dip down into the medulla. So the renal tubule is made up again of well, first we have the Bowman's capsule, or the glomerular capsule. And then we have the proximal convoluted tubule. Convoluted means just all random, crazy. Here we're going to deal with reabsorption and secretion. Right? I'm going to define what these terms mean here. And these are in the cortex area, the cortical area. Loop of Henle descends into the medulla and then ascends back up as well. And then the distal convoluted tubule, this one will function more in secretion than in reabsorption. And I'll explain what that means. And it's confined to the cortex. Collecting ducts receive filtrate from many nephrons. They fuse together to form the minor calyx or minor calyces. And they're going to then help to move that fluid along to the major calyx, to the renal pelvis, and then finally out the ureter. So again, just to give vocabulary here, location. We are at the cortex medulla junction, right? So that the watermelon shell, peel, whatever you want to call it, and then the red part, right? So that junction between cortex and medulla. So in the cortex, we have the glomerulus, glomerular capsule, Bowman's capsule, proximal convoluted tubule. The loop of Henle extends down into the medulla and then back up into the cortex, where we have then the distal convoluted tubule, and then our collecting duct. All of these little cut areas represent different distal convoluted tubules that are feeding into this collecting duct there. So most of the nephrons, about 85% of the nephrons, are cortical nephrons. They sort of are in that junction of the cortex and the loop of Henle going down into the medulla. There are different types of nephrons, a smaller number. We call them juxtamedullary nephrons. So they're gonna they're gonna kind of protrude very deep down into the medulla there, right? So here's the first one we're talking about. So we have our glomerulus surrounded by the glomerular capsule, the proximal convoluted tubule, the loop of Henle, the ascending loop of Henle, right? So it's coming this way. So we're descending down, ascending back up, and then our distal convoluted tubule, which connects to our collecting duct. Now, if you notice, this nephron is sitting more superiorly than is this nephron. So here's our juxtamedullary nephron. So it sits lower, still in the cortex. The proximal convoluted tubule is still in the cortex, but the loop of Henle dives much deeper into the medulla. And then again, our distal convoluted tubule to our collecting duct. So I hope you see they are different, right? We have these, what we call peritubular capillaries in purple, arranged differently. Again, form fits function. We know that they're going to both filter, but they're going to have different sort of final products, final functions for the body that they're going to accomplish later. So looking at our glomerulus, right? If we follow circulation, right? We leave the aorta, we're arterial. We shift into now the arterioles. We have a capillary bed that we call the glomerulus. And we go now to efferent arterioles. So the capillaries of the glomerulus are both fed and drained by the arterial system, right? The arterial, oxygenated blood going away from the heart, right? So we're still going away from the heart, still going away from the heart. Then we enter into this purple area, which we call peritubular capillaries. Perimeter, they're around the tubules, and we call vasa recta. The vasa recta are going to be part of our juxtamedullary capillaries. Now we see a change in color from red to purple to blue. These peritubular capillaries, vasa recta, are normal, everyday, boring type of capillaries. They do what all the other capillaries do. They start arterial. We have gas exchange, we have nutrient exchange, and then we start to work our way back towards the heart. So we go back in blue now, going to the veins, right? To the vena cava, back to the right atrium. So the whole concept here, the glomerulus is unique. These capillaries are a higher pressure capillary bed than are these capillaries. These glomerular capillaries are designed to force fluid out, to force plasma to become filtrate in both types of nephrons, I should say, but in the capsule area, right? So we're forcing high pressure blood into the tubule system of the nephron, and then we're going to kind of pick stuff back up here at the peritubular. So again, if you notice here in purple, the peritubular capillaries, very highly enmeshed around the convoluted tubules. Vasa recta looks like a net. Looks like those cargo nets that they climb. It's very, seems more structured. It's not as random and chaotic over here. Right? And there's a purpose for this. So when we go back and link now cardiac output, right? Cardiac output, renal arteries deliver about 1,200 mils of cardiac output to liters each minute. So 1.2 liters. And we have about 5 liters of blood. So 1.2 liters of blood is coming to the kidney each minute. So that tells you these kidneys are working hard. They're filtering a lot here. So the kidney filters the body's entire plasma volume 60 times each day, right? So that's a lot. Like the whole volume of blood that we have gets filtered around 60 times per day. Filtrate is blood plasma minus proteins. So the glomerular capillaries are basically able to leak out plasma, but they're not able to leak out proteins. They're not able to leak out antibodies or other proteins, albumin, that kind of stuff. So the urine itself will be less than 1% of the total filtrate. So that's important, right? So if we're filtering this much blood, if this much fluid is being pushed out into these tubules, that means that if we were to urinate out that much amount of volume, we would dehydrate and die very rapidly. But again, less than 1% of the total filtrate is going to be excreted out. So in order to understand this, we need to kind of define some vocabulary terms here, right? Talking about filtration, reabsorption, and secretion. So filtration, reabsorption, and secretion. So this is going to be a matter of perspective, where we're standing, right? So if I am, let's say, in this room, and I walk out the door, do I say, am I walking out of the room, or am I walking into the hallway? Well, technically, they're both right? So I'm walking out of the room, but I'm walking into the hallway. So it just depends. How can I be walking out of something and into something at the same time? Right? Well, it's just a matter of perspective. So glomerular filtration, we are going to leave we're leaving the circulatory system. I'm forcing fluid out of the circulatory system. Reabsorption, oh, actually I need that. Can I have that back? So I'm reabsorbing what was lost the first time, right? So we leak out a bunch of fluid. Tubular reabsorption, I reclaim glucose, amino acids, 99% of the water, salt, other components back to the blood. Right? And in this situation, in tubular secretion, reverse of reabsorption, selective addition to the urine. So we have a couple of things that are happening here, right? One, the blood is forcing out all this liquid, plasma converting into filtrate. Now within the nephron, the nephron says, no, well, this is good, this is good, this is good. Let's push this back into the blood, right? So a lot of that gets pushed back into the blood. And then the blood says, oh, you know what? No, I don't want this, right? So it's excreted back out. So if we can make sense of this, hopefully you understand this process. So one, right, glomerular filtration. So this is when we push fluid out of the blood vessel into the capsule. So we're in here, and now we have what we call tubular reabsorption, 2. So the good stuff is returned back to the circulatory system. And then 3, no, we don't actually need that. So that is pushed back into the tubular system, so it can be excreted as urine. So this is the way that filtration happens at the nephron level, right? This microscopic level. This always reminds me of garage sales. I don't know if you ever had garage sales or you sell stuff on eBay or whatever. A garage sale, right? You put stuff out and then, oh, you know what? I remember that toy, man, sentimental value. I don't want to get rid of that toy. I'm going to take it off the table and I'm going to keep the toy. And then, well, you know what? I don't really need the toy. I don't have that much sentimental value to it. Let me put it back out on the table. So that's kind of what's happening. So the stuff is sort of released out. That's filtrate. Filtrate then can be reabsorbed when I recollect some of this stuff back. and then, you know what, I'm going to be honest with myself, I don't really need it, I'm not going to use it, then it gets placed back on the table. And that's, again, that secretion again. So again, lots of stuff happening. So we have afferent, efferent circulation. Glomerular high pressure forces plasma minus the proteins to enter into the capillary, right? Into, not the capillary, that we leave the capillary into the Bowman's capsule. Glomerular capsule, that is step one, that's filtration. We're filtering. Two, well, we want to keep some of that stuff, so it's going to be reabsorbed by the blood vessels, by the peritubular capillaries, or by the vasa recta. And then we changed our mind. We're actually going to go ahead and secrete this out, and that then three. And at this point, if you're stuck in here, there's only one way to go now, right? At this point, all we can do is be excreted out. So that's the roles of our nephron, basically, yeah? So glomerular filtration, just understand, it's a high pressure system. For capillaries, that's strange. We have a high pressure system that's forcing out a lot of the fluid. Our filter is about, 5 nanometers. Molecules that are greater than 5 nanometers are not filtered. So the large proteins cannot be pushed out. If somebody's urinating a lot of proteins, we say, well, that's not quite good. There's some issue, some problem where this limit is being exceeded, right? So proteins are much larger. They should not be found in high amounts in the urine. I'm not going to get into all this. We would have done all this stuff in lab. Calculating pressures and same concept as blood flow. We're always going to follow high pressure to low pressure areas. Fluids work like this, gases work like this, the kidneys no different, right? We're always moving from high pressure areas down to low pressure. Glomerular filtration rate, again, we can theoretically analyze it. Basically, the volume of filtrate formed by minute. So we're looking at about 120 to 125 mils per minute is an average for a healthy individual. Let's see if I can get to the rest of this part. Right, so we've worked our way to the nephron. Now let me look for the next discussion that takes us from the nephron down to the bladder, and then sort of the urethra, right? So let me pause that here.